Welcome to my channel. Today I will discuss about ribozyme. Ribozyme. Comes from RNA, that is ribonucleotide, and zyme comes from enzyme, that is ribozyme. So ribozyme, from the definition, you can see ribozyme is an RNA molecules. Bear in mind they are RNA molecules, but they act as a biological catalyst, similar like enzymes, like protein enzymes, and they can perform different type of chemical reaction, and they bind with the substrate, make the product something exactly like the proteinogenic enzymes, but they are non-protein enzymes. In 1980s, Sidney Altman and Thomas Cech, they discovered ribozyme, and for this they got Nobel Prize in 1989. So after the discovery of this ribozyme, it's give a strong support for RNA World Hypothesis, because they showed that RNA has the capability to catalysis, and they have the both genetic material and catalysis. Let's talk about RNA World Hypothesis. RNA World Hypothesis is possessed that early life on Earth was based primarily on RNA molecules before DNA and proteins evolved. That's the RNA World Hypothesis. So RNA could have served as dual role. First, they can store the genetic material, something like DNA, and also they have the ability to biocatalyst like enzymes. These are the RNA World Hypothesis. So Cech and Altman, actually they showed for the very beginning that RNA has the capability to catalyst, which support this RNA World Hypothesis. And later on, this RNA World Hypothesis shifted to DNA World Hypothesis or DNA Protein Hypothesis, because later protein emerged as better catalyst, and also DNA emerged as the more stable storage molecules than RNA. As we know that DNA lack 2' OH group, which is that's why it's less hydrolysis than RNA. That's why DNA is chemically more stable than RNA, and it also allows error correction by proofreading, and also support larger genomes. That's why DNA world hypothesis is most prominent at this moment. And also enzyme like ribonucleotide reductase, which help to convert RNA nucleotide to DNA nucleotide. That means RNA can also be converted into DNA. And then once DNA become the genetic materials, then central dogma emerged. Like we know, central dogma is from DNA to mRNA, mRNA to protein. Here DNA played a vital role, like storage of genetic material, and RNA played vital role in a messenger or regulator, and in case of protein, catalyzed. That's all about RNA World Hypothesis. Let's talk about the timeline of ribozyme discoveries. It's actually from 1967, while Carl Woese, Francis Crick, and Lesail Orgel first they proposed that that RNA could process catalytic capabilities, but they can't find any experimental evidence for this statement. But they believed that RNA could process catalytical capabilities. So that's lying the conceptual foundation for ribozymes discovery. In 1982, both Thomas R. Cech from University of Colorado discovered that a ribosomal RNA precursor in tetrahymena, they have the capability to self-excise an intron in vitro without any help of any protein. That means that demonstrating that RNA has the capability to catalyze like enzyme. At the same year, in 1982, Sidney Altman from the Yale University, they also showed that RNA component of E. coli, that is the RNase P. RNase P has two part, protein part and RNA part. This RNA part have the capability to process tRNA precursor without any protein factors. So they proved that RNA can have the capability like the enzyme to catalyze different type of biochemical reaction. For these discoveries, wonderful discoveries, they got Nobel Prize in 1989. And from that time to now, a lot of in vitro evaluation related to ribozyme discovered at this moment, and among them Hammerhead ribozyme, Hairpin ribozyme, catalytic RNA within the ribosome, that is the peptidal transferases activity are discovered, and also a lot of artificial ribozyme introduced to study different type of genetic therapy or gene studies or genomic studies. Let's talk about major types of ribozyme. We can classify ribozyme like self-splicing introns, ribosomal RNA, RNase P, self-cleaving ribozymes. First, let's talk about self-splicing introns. Self-splicing intron we can classify in two groups: group 1 introns and group 2 introns. In group 1 introns, they remove the introns from RNA by splicing. That means RNA themselves, introns themselves can be spliced from the RNA by using a free guanosine as a cofactor. So they need a free guanosine, and also they need magnesium ion. I will discuss it later. And in case of group 2 introns, they self-splicing by using an internal adenosine. So they use adenosine. And then in case of ribosomal RNA, which it makes the ribosome a giant ribozyme. That means ribosomal RNA itself a giant ribozyme, because it contains peptidal transferase center which catalyze peptide bond formation, like in the large ribosomal subunit like 23S RNA in prokaryotic and 28S RNA in eukaryotic. And RNase P, which I told earlier that it a ribozyme that process tRNA precursor. How it process it? It actually cleaving the extra nucleotide in the 5' part of the tRNA. I will show it later. And self-cleaving ribozyme, which we called small ribozymes. They actually widely found in plant viroids, viruses, and small cellular RNAs. For example, Hammerhead ribozyme, Hairpin ribozyme, hepatitis delta virus ribozyme, and GLMS ribozyme. Here, Hammerhead ribozyme is found mostly in plant viroids and satellite RNA, whereas Hairpin ribozyme found mostly in tobacco ring spot virus satellite RNA, and hepatitis delta virus, this HDV ribozyme found mostly in human pathogen HDV, and GLMS ribozyme, which found mostly in bacteria, which function as a riboswitch, and it cleaves itself upon binding glucosamine 6 phosphate. Let's talk about major types of ribozyme. Again, we can classify most broadly in natural and artificial. In natural also we can classify in two types, like small and larger ribozyme. In small group we can classify into Hammerhead ribozyme, Hairpin, hepatitis delta ribozyme, which we called HDB, and Varcoed satellite. And in case of large, like group 1 introns, group 2 introns, and also RNA component of RNase P, these three we can classify into large group of ribozyme. And here are the 2D structure of this ribozyme. First one, Hammerhead ribozyme and Hairpin ribozyme, looks like this in 2D structure. HDV ribozyme and group 1 ribozyme here, and group 2 ribozyme look like this, and RNase P ribozyme look like this, and they act on the extracellular part of tRNA. I will discuss it later. Let's talk about Thomas R. Cech's discovery. Thomas R. Cech discovered in 1982, as I told, that certain introns, that is the non-coding sequence of RNA, could splice themselves. They can splice themselves without any help of any protein, and this phenomenon known as self-splicing. Bear in mind, self-splicing and self-cleaving, both are not same, a little bit difference. And what it need actually, it need RNA itself and magnesium ion as cofactor, and free guanosine nucleotide also. It act as a cofactor, is a metal ion, and this guanosine nucleotide play a vital role for initiating this enzymes activity, ribozymes activity. So what actually they found? They found that after painstakingly removing all protein part from the experiment, they exclude all the protein part, and they were stunted to find that RNA still managed to splice itself. That means they find that some protein part are there. So what they did, they first use detergent to denature this protein, and then use protease enzyme to vanish this protein, and they did it nicely. And but still they found that the splicing of intron splicing is occurring by itself. So they proved that RNA has the capability to act like an enzyme. And here, as I told, guanosine nucleotide act as a essential cofactor, and that initial the attacking of molecule as a nucleophile. Let's talk about the group 1 intron. As group 1 intron, we know group 1 intron and group 2 intron, these are self splicing group of ribozyme. So I will discuss about group 1 intron. Group 1 intron has two-step process to trans esterification. Let's talk about the group 1 intron, which I has told that it has two-step process involving the transfer of phosphodiester bond. Here in first trans esterification process, what happened actually, in first trans esterification process, a free guanosine molecules or a related guanosine nucleotide like GTP, it's bind to a specific site within the folded intron, and which function as a ribozyme. And then what happened, then this 3' hydroxyl group of guanosine, this 3' guanosine, this OH is attached at the 3' part of guanosine, and then this guanosine initiate a nucleophilic attack. Where it goes, then attack nucleophilic attack on the phosphodiester bond. This is the phosphodiester bond at the 5' end of exon. So this is the 5' end, this is 5' end, and this is 3' end, bear in mind. And 3' hydroxyl group of guanosine, don't be confused about it. So what happened, first this guanosine attack on the phosphodiester bond of 5' part of exon, and then what happened, then this cut the RNA, then this cut the RNA here, and still guanosine attached in being covalently linked, guanosine covalently linked with the 5' intron. This is the 5' intron, and this is 3' intron, bear in mind. So in 5' part of exon still contain 3' OH of guanosine, and then second step come. Then second step how come, in second transesterification process, and this OH group, 3' OH group of guanosine, which stay with the 5' part of exon, attack here, attack the 3' part of the exon. This one is exon 3' part, here is 3' as I told. And then what happened, then they kick out this intron, and then they ligate this exon with this exon within phosphodiester bond. And later this free intron lariat form a lariat form, that is a circular form, and after that a enzyme called DBR1, that is the lariat debranching enzyme, act on here to make it unbranch, and also to use for further recycling process. That's all about group 1 intron. I can explain again how it actually work. Here, as I told, guanosine molecules first bind to the specific site within the folded intron. This first bind to the specific site, so this one is the specific site, for example. Then this guanosine of 3' hydroxyl guanosine group then initiate the attack, nucleophilic attack on the junction of intron and exon. This is the 5' intron exon, this is 5' intron, this is 3' intron, this is 3' exon, bear in mind. Then what happened, then it cut here, but still this guanosine covalently attached with the 5' end of intron, and in case of here the covalently attached with the 5' part of intron, but still the OH group, which OH group, it comes from 3' guanosine OH group. This 3' guanosine OH group attached with the 5' exon, and then this OH free OH attack 3' exon. This one is 3' exon, as I told earlier. Attack here, and then the intron became released, and also it attached with this exon part, attached with 5' exon part, attached with 3' exon part, and then this free intron make lariat, that means it makes circular, and after that an enzyme called debranching enzyme or DBR1 enzyme, it acts on here and make it unbranching and ready for another use or recycling. Let's talk about RNase P. In Altman's, it's actually Altman's discovery. Altman's find that RNase P, and he find RNase P has two parts. One is protein part and another one is RNA part. But this protein part is not involved in catalytical activity, where RNA is involved in the catalytical activity. So he showed that in his experiment that this RNA has the capability like the to catalyze like the enzyme, and this RNase P process the precursor tRNA. So these are the tRNA, and tRNA, some extra part of this tRNA cleaved, and by cleaving this they become the mature tRNA. And then Altman showed that RNA subunit alone, that is the RNA subunit alone without the help of protein, could catalyze the cleavage reaction. Then you may ask that what the function of this protein? This protein actually stabilize these RNA molecules. Let's talk about Altman's discovery. In Altman's discovery, the overall cutting process involved three key steps. First one is recognition, second one is activation, and third one is catalysis. Let's talk about recognition. How recognize? RNase P bind to the pre-tRNA substrate through a dual anchor mechanism. Here RNase P bind with the premature tRNA in dual anchor mechanism. Then what's actually dual anchor mechanism means? His first anchor is the acceptor stem or T stem recognition. This one is acceptor stem or T stem. T stem recognition. First RNase P contact with T stem loop or T stem loop, and then ensure correct tRNA sequence. That means this one is to ensure that this is correct tRNA and the shape is okay, universal. And then second anchor is occur with the 5' leader sequence recognition. That means, and second it recognize 5' end of leader sequence. Then what it did? 5' end recognition. RNase P interact with the 5' leader sequences. RNase P interact here, and this helps to direct the recognition size. For example, this one is recognition size site. Then they direct here the L-shaped. This one is L-shaped folded structure of the tRNA, where this RNase P bind with this junction and it form a ES complex, that is enzyme substrate complex. It form here, and this ES complex is irreversible. And then activation come. How activation occur? Then magnesium ion bind here to activate it in a enzyme state activation, enzyme substrate activation. Then it activate, and then catalysis occur. How catalysis occur? Just here cut one nucleotide. Bear in mind it cut only one single endonucleotide cut at the junction, and then hydrolyze the photodiester bond. It hydrolyzes this photodiester bond, and then 5' later release. And again, in case of mature tRNA, still a monophosphate bond occur here. A monophosphate bond still remain with the mature tRNA. And then what happened in 3' end of tRNA? Here you see RNase D work here. RNase D. Bear in mind RNase D is not ribozyme. It helps to trim any nucleotide, a single nucleotide it can trim. For example, here it trim G and to add the CCA end. You know, CCA end is the universal end of any tRNA. So here only G reduced. Of course, if it need, RNase D can cut also 2 or 3, but they will do it one after another, not 3 or 4 at a time, one at a time. So this one is the mature tRNA. That's how RNase P helps to convert pre-tRNA to tRNA, mature tRNA. Let's talk about hammerhead ribozyme. Why it's called hammerhead ribozyme? Because its 2D structure look like the hammerhead shark, and it contain 3 stem 1, 2 and 3, stem 1, 2 and 3, and it has a catalytic core around 15. It has a catalytic core around 15 nucleotide conserved nucleotide. It may be changed, and also it need magnesium ion or any other divalent cation. You know, magnesium ion act as a Lewis acid. What Lewis acid is? It's actually an electron pair acceptor that stabilize the negative charge on the phosphate background during the transition state. And in other case of magnesium, we can use any divalent cation like manganese. And then here the cleavage occur at a conserved domain NUH. N is any nucleotide. It may be any nucleotide, N, U and H. So here cut. So this is the cutting point of hammerhead ribozyme. This is self-cleaving RNA motif. And let's talk about the function of ribozyme. So far we have seen that ribozyme performed a lot of functions like RNA splicing, introns, RNA cleavage and ligations. We have seen it cut and join RNA molecules, and also peptide bond formation, like in rRNA it form peptide bond. And then RNA processing, it helps to process the RNA like tRNA maturation, rRNA processing like peptidal transferase activity, which help to form peptide bond. Let's talk about the characteristics of ribozymes. RNA first is RNA-based catalysis. This our ribozyme is RNA-based, not protein. That is non-protein part, but they can catalyze exactly like the proteinogenic enzymes. And their catalytic activity, they can cleave, ligate, splice or polymerize RNA. And their specificity, they are very much specific like protein enzyme. They also have substrate, and they make the substrate enzyme complex, and they have self-splicing ability, as I told earlier. And they also perform peptidal transferases activity like ribosome. It's a giant ribozyme, rRNA, form peptide bonds. And then evidence of RNA world, as I told earlier, that in RNA world it was believed that RNA perform two dual role like genetic material reservoir and catalytic activity. So in this ribozyme discovery, it proved that RNA world hypothesis was correct. And then, but most importantly, it require cofactor magnesium ion. Bear in mind, ribozyme need cofactor like magnesium. Now let's talk about the summary of today's lecture. First, ribozyme is an RNA molecules that act as biological catalyst, and it's non-protein enzyme. And RNA world hypothesis, what RNA world hypothesis is, early life of Earth was based primarily of RNA molecules before DNA and protein evolved, and it played dual role. RNA played dual role as genetic material and catalyst. This is the RNA world hypothesis. And major type of ribozymes, we can classify ribozyme broadly in natural and artificial. In natural, we can classify broadly in large group and small group. In large group, it's in large ribozyme group 1 and group 2 intron and RNase P. And in case of small ribozyme like hammerhead ribozyme, hairpin ribozyme, hepatitis delta virus ribozyme, GLM S ribozyme, these are the small ribozyme. And let's talk about Thomas Cech's discovery. That is the group 1 intron. As I told, group 1 intron, it involved in 2 steps. First, the transfer, 2 steps involved to the transfer of phosphoester bond. First, guanosine attack the junction of exon and intron, and then it release that 5' attacked with the intron, and then 5' exon where OH was attached there, and it attacked the 3' part of exon, and then it released the intron and then ligase. This is the overall function of group 1. And then Altman's discovery, that is the RNase P, which is the RNA contain 2 part, protein part and RNA part. RNase P contain 2 part, RNA and protein part. Here RNA part acts as catalyst and protein part helps to stabilize this, and it involved in 3-step recognition, activation and also catalysis. Recognition, RNase P bind to the pre-tRNA substrate through a dual anchor mechanism, and first anchor is it recognize acceptor stem, and second anchor is it recognize 5' leader sequence to direct exact the junction, L junction of premature tRNA. And then magnesium add here to activate the ES complex, that is substrate enzyme complex, and then catalysis occur by cutting one nucleotide and then release 5' leader sequence, and also attached P with attached a phosphate group with the mature tRNA. That's all about today's lecture. Thank you very much.